Hi guys, welcome back to the Science National Honor Society lecturing series. Today we're going to be going over Chapter 5, which is the working cell. And the essential question for this chapter is how does the cell membrane function? And what exactly goes on within the cell membrane and what components make it up that give it its specific structure and function. So basically there's three main things that you should know about the cell membrane before we move along. It is said to be a fluid mosaic model, it's selectively permeable, and the functions of the proteins in the plasma membrane allow many different particles to get inside of the cell. So a fluid mosaic model basically says that the plasma membrane is a phospholipid bilayer and it has many different proteins embedded in it. Now in this fluid mosaic model, you're going to have hydrophobic heads that are fatty acid tails on the inside, and the outside is going to be hydrophilic heads. Now, the hydrophilic heads are going to be permeable to water and many other polar molecules, whereas the inside of the fatty acid tails, they're not going to be permeable to water because it will be more permeable to nonpolar molecules. Now, this membrane is also selectively permeable because it regulates what can go inside and outside of the cell, and its selective permeability can be attributed to the fact that it has both a hydrophobic and a hydrophilic region. Now, in order for different hydrophilic molecules and polar molecules to get through the plasma membrane and inside of the cell, they're going to need many different proteins to help them do this. So this goes into the process of passive transport and active transport, which we will take a look at. So here we can see a diagram of what the cell membrane actually looks like. So as you can see here, these yellow strands are going to be the fatty acid tails, and this is the hydrophobic interior of the plasma membrane. And these purple dots on the outside are the hydrophobic heads. So what this means is that if something is hydrophilic on the outside, then it is permeable to things such as water. But on the inside, since it's hydrophobic on the inside, water and other polar molecules aren't going to be able to pass by. And that's why we have all these different proteins embedded within the membrane, because these other molecules that can't necessarily get in on their own still need to find a way to get in. So they can get in through various ways, such as through signal transduction pathways, active transport, passive transport, facilitated diffusion, and cell-to-cell recognition. Now, the formation of this membrane is really unique because phospholipids were among the first organic molecules created, and this membrane is very important because it allows the cell to regulate chemical exchanges with its environment. Now, because organisms are open systems, in order to survive, they need to communicate with their environment to make sure that they have all of the proper nutrients and growth factors and everything else that they need to survive, which is why these cells need to regulate exactly what's going inside of them and what's going outside of them so that they can function. Now here we're going to be getting into passive transport and diffusion. So diffusion is the movement of materials across a membrane down their concentration gradient. And in diffusion and passive transport, there is no expend of energy. So in order to go through diffusion and have passive transport, you're not going to need anything like ATP or any energy to facilitate this process. And this is because all these reactions occur because of thermal energy, and this is because of the constant movement that exists between solute particles. Now, these solute particles are going to move until they are in a state of dynamic equilibrium, which is shown here. Now, in this picture, we can see that there is a higher concentration of solutes here, and there is no concentration of solutes here. And there is a lower concentration of water here than it is here. Now, these particles are going to diffuse across the membrane back and forth until they meet a state of dynamic equilibrium. Now, equilibrium is not to say that the movement between these two sides will halt, but what it means is that there will be no net gain or loss between the two sides, but the particles will still be continuously exchanging. Now, when speaking of diffusion, the diffusion of water across a membrane specifically is osmosis. Now, this is a type of passive transport, which means it also doesn't require any energy. And because water is a polar molecule, we learned that the interior of the plasma membrane is hydrophobic. So if water wished to pass through the interior of the membrane, it would be extremely difficult, but cells need that water to survive. So they're going to use proteins called aquaporins, which are embedded within the membranes, to allow themselves to transport themselves inside of the cell. Now, in this example, which is called the U-tube example, this is a very, very common diagram or picture of what you would see to kind of display osmosis. So as you can see here, on this side, we have less solute particles than we have here. This is the higher concentration, and this is the lower concentration. However, on the side with less solute particles, you're going to have a higher concentration of water, and vice versa on the other side. Now we know that water moves from high to low, so if there's a higher concentration of water here, it's going to move in this direction. However, for solute particles, it's the opposite. So solutes move from areas of lower concentration to areas of higher concentration. So because I have a lower concentration of solute particles here, it's going to move across the membrane onto this side. Now, when speaking of water balance between cells, we often refer to their tonicity, and this can be seen in three key ways. You can have an isotonic environment, a hypotonic environment, as well as a hypertonic environment. Now, in an isotonic solution, there is essentially dynamic equilibrium between the concentration inside of the cell and outside of the cell. Now, this goes for both the water concentration and the solute concentration. However, in hypotonic solutions, you're usually going to have a cell that is lysed because of the environment that it is in. Now, in hypotonic solutions, the concentration of solutes on the outside is lower than what is on the inside, and the concentration of water on the outside is higher than what's on the inside. So because we have a lower solute concentration and a higher water concentration on the outside of the cell, both of these things are going to work their way inside of the actual cell, causing the cell to swell up and eventually burst, which means that it will lyse. And the way that I like to remember this is that the word hypotonic has an O in it. So if you imagine this O as the cell, you know that this O is going to eventually lyse and burst because of everything that's flowing inwards. However, in a hypertonic solution, it is essentially the exact opposite because the cell will shrivel up. Now, this is because the concentration of solutes on the outside is going to be higher in relation to the concentration on the inside. So, the solute particles on the inside of the cell will then move outwards into the environment. And the water concentration is going to be higher on the inside than it is on the outside, so the water will move outwards because water also moves from areas of high concentration to low concentration. Now, in animal cells, this will lead the cell to become shriveled up because if you think about it, if the solute particles and the water particles are exiting, then your balloon or your cell is essentially becoming deflated and it's going to become shriveled up. However, in plant cells, there is something called plasmolysis that is used to describe this phenomenon. Now, because plant cells have a cell wall, their reaction to hypertonic solutions is a little bit different because their plasma membrane is actually going to detach from the cell wall after it has become shriveled up, and because of this disconnect, the cell will ultimately shrivel up and die. Now here we can see an example of facilitated diffusion. So hydrophilic molecules and ions that need help crossing the plasma membrane use specialized transport proteins. Now an example of a specialized transport protein that we mentioned before was the aquaporin. So if we have a certain molecule that needs to make its way inside of the cell, but it's a polar molecule or it's too big to pass through the membrane alone, it will use facilitated diffusion to do so. Now here we have protein channels, and every single type of protein that you see inside of the membrane is specialized and has a specificity to a certain type of molecule. So in this protein channel that we see over here, a certain type of protein will be brought through the channel so that this protein can make its way inside of the cell. And in order for this process to happen, we don't need any expend of energy. In carrier proteins, these molecules will make their way and attach to a carrier protein, and this protein will see a conformational change and then release those carrier proteins to the inside of the cell. So here we're going to be taking a look at active transport. Now, active transport is the movement of substances across the plasma membrane against the concentration gradient, and this uses ATP and cellular energy. Now, this is going to be the major difference between active transport and passive transport. Now, the key example of active transport is going to be seen as the sodium-potassium pump and nerve impulses. Now, as you can see here, you can tell that this is an example of active transport solely because you can see that ATP is being used to help out this reaction and allow it to go. Now in this diagram, we can see the sodium-potassium pump. So here, cytoplasmic sodium is going to bind to the sodium-potassium pump, and on the outside in the extracellular fluid you have a high concentration of sodium and a low concentration of potassium, and on the inside of the cytoplasm you have a low concentration of sodium and a high concentration of potassium. Now whenever sodium binds to this protein, it stimulates phosphorylation by ATP. Now, a phosphate group will then bind to the protein, which will release the sodium ions outwards. Now, in order to get this protein back to its original state, the extracellular potassium is going to bind to the protein, and this triggers the release of the phosphate group. Now, since this phosphate group has been released, the potassium particles as well will get inside of the cell, but now this protein is back to its original state because that phosphate that was originally attached here is now gone. Now, this is an example of active transport because none of this could have happened if this ATP was not present here. Now, endocytosis and exocytosis are seen as ways that cells can gather food from themselves, and these two processes are essentially exact opposites from each other. Now, what exocytosis is, it's how the cell will release their contents, because in exocytosis, transport vesicles will migrate to the membrane and fuse with it, and this will allow them to release their contents. Now, many secretory cells use exocytosis to export their products, and this can be seen here because the product is on the inside and it will ultimately move out into the extracellular fluid. Now, endocytosis is different because here, the cell is going to take in macromolecules by forming vesicles from the plasma membrane. Now, endocytosis is the reversal of exocytosis, and it involves many different proteins. Now, there's three main types of endocytosis, and this is phagocytosis, pinocytosis, and receptor-mediated endocytosis. Now, phagocytosis, I like to think of it as cellular eating, and pinocytosis is like cellular drinking. Now in this diagram, we can see three examples of phagocytosis, pinocytosis, and receptor-mediated endocytosis. So in phagocytosis, which we have here, a cell will engulf a particle into a vacuole, and that vacuole will then fuse with a lysosome to digest this particle. In pinocytosis, molecules are taken up when the extracellular fluid is gulped into tiny vesicles. So here we have the fluid, and it's ultimately going to be taken up here, and a vesicle will be formed to allow this fluid to enter the actual cell. Now, that's how I like to differentiate them, because this is, like I said before, this can be seen as the drinking, and this can be seen as the eating. In receptor-mediated endocytosis, the binding of ligands to receptors triggers vesicle formations. Now, what a vesicle is this, what we have right here, and a ligand is any molecule that binds specifically to a receptor site of another molecule.